Number 1. Vulcanoctopus hydrothermalis, the vent octopus. 2,500 meters beneath the surface of the eastern Pacific, the ocean floor splits open. Black smoker chimneys rise from the basalt like industrial stacks, blasting superheated water at 350 degrees Celsius into the surrounding darkness. The fluid is loaded with hydrogen sulfide, dissolved metals, and particles so dense that visibility drops to almost nothing. The temperature swings hundreds of degrees across a distance you could cover with your hand. Nothing should be alive here. But crawling across the base of one of those chimneys, tentacles splayed over jagged mineral rock, is a pale, translucent octopus, Vulcanoctopus hydrothermalis, the only octopus on Earth that lives directly inside an active hydrothermal vent field. Gonzalez and colleagues described the species in 1998 from specimens collected at the East Pacific Rise. It looked wrong from the start. When they dissected it, they found something no one had seen in any insurate octopus before. The ink sac was gone. Not reduced, not vestigial. Completely absent. Every other octopus in its lineage has one. This species evolved it away. And the reason is straightforward. Ink is a visual defense. You squirt it, you disappear behind a dark cloud while a predator loses track of you. But in the vent field, the water is already so saturated with mineral particles that visibility is effectively zero. Squirting ink into that is like throwing smoke into fog. It's useless. So evolution removed it. What replaced vision is chemistry. The octopus hunts by chemoreception and touch, detecting the chemical signatures of vent amphipods and polychaete worms on the chimney surface. Its eyes are reduced but still functional, not for seeing shapes or prey, but for detecting the faint thermal glow that superheated fluids emit above 350 degrees. That glow is invisible to the human eye. The octopus can see it. It uses heat as a landmark. Its blood tells the rest of the story. The hemocyanin, the copper-based oxygen-carrying protein in its blood, is specifically adapted for binding oxygen at the vanishingly low levels found in vent water. Most marine invertebrates would die outright from the hydrogen sulfide concentrations this octopus tolerates routinely. Its metabolic tolerance is not a passive accident. The hemocyanin molecule itself has been restructured to function under conditions that would denature its counterparts in shallow water cephalopods. Females brew their eggs directly on the chimney walls, exposing embryos to sulfide-laced water from the first moment of development. That means the offspring aren't just adapted to vents. They are developing inside one. The mantle musculature is thinner than in shallow water relatives, reflecting a low-energy existence in a food-limited environment. Its radula, the feeding structure, is modified for scraping rather than piercing. In some contexts, this octopus is as much a scavenger as it is a predator. It lacks the chromatophore complexity of its shallow cousins. The skin is uniformly pale because camouflage is pointless when nothing can see you. It is an organism that has been simplified by darkness and chemistry, stripped down, rebuilt for a world where the rules are different. No one knows how many exist. Each vent field may support only dozens of individuals, and when a vent field goes inactive, which happens unpredictably, sometimes within decades, the entire local population presumably dies. No migration between vent systems has ever been documented. The octopuses at one chimney cluster cannot reach the next. They are marooned. Specimens from two different vent sites on the East Pacific Rise, 13 degrees north and 9 degrees north, show slight morphological variation. That variation is the fingerprint of populations beginning to diverge. The split from the species' nearest relatives is estimated at 30 to 40 million years ago by molecular clock analysis, but the divergence between its own populations may be much more recent. And it is still happening. Originally placed within the genus Bythograeid by Strugnell and colleagues in 2008, it was later reassigned to its own genus because the morphological differences were too significant to ignore. It coexists with Bythograeid crabs and Riftia tube worms in the vent community. We will meet both of those later. But the vent octopus holds a niche no other cephalopod has ever claimed. A predator operating in total chemical darkness, at temperatures that swing from near freezing to scalding within centimeters, in water that poisons most life on contact. Evolution didn't just adapt it, it redesigned it from the ground up. If losing your ink sac sounds like a drastic evolutionary trade, The next creature gave up something even harder to explain. Its tentacles detach from its body, and they keep hunting on their own. Number 2. Grimalditeuthis bonplandi, the phantom tentacle squid. Leave the vent field, rise through 1,000 meters of black water. There's nothing here. No floor, no ceiling, no walls. The mesopelagic and bathypelagic zones of the open ocean are the largest habitat on the planet by volume, and they are almost completely empty. Prey density is so low that a predator can go days or weeks without encountering a meal. Chasing food costs energy you don't have. So how do you eat? Grimalditeuthis bonplandi solved the problem in a way that no other cephalopod has ever attempted. It sends its tentacles out to fish for it. The tentacle clubs, the flattened pads at the end of the feeding tentacles, detach or extend far from the body and begin to undulate on their own. They perform rhythmic sinusoidal waves. They flutter and ripple through the water column, mimicking the movement of a small planktonic organism. Anything that comes to investigate the motion swims straight into the squid's waiting arms. Augustin Vergny first described the species in 1837, but no one understood its hunting behavior for another 176 years. When researchers collected specimens, the tentacle clubs had no suckers and no hooks. Every other squid on Earth has them. The clubs looked damaged. Scientists assumed they were degraded during trawling. It was not until 2013 when Hoving and colleagues at MBARI published ROV footage from 1,500 meters in Monterey Bay that anyone saw what was actually happening. The clubs were not broken, they were bait. The squid's body stays nearly motionless during the hunt. It hangs in the water column, conserving every calorie while its detached tentacle clubs do the work. The energy cost is almost nothing. The club tissue contains elastic proteins that enable passive wave propagation. The undulation is partly self-sustaining once initiated, like a flag in the wind. Bioluminescent photophores on the clubs may enhance the lure in total darkness. The squid is using aggressive mimicry through an autonomous body part. Nothing else in the cephalopod lineage does this. Nothing else in any lineage does this. Its body confirms the strategy: weak musculature, minimal mantle thickness, a build designed for ambush, not pursuit. Stomach content analysis reveals a diet of small fish and crustaceans, exactly the kind of organisms that would investigate a wriggling planktonic shape in an otherwise featureless void. It is the sole member of its genus, sitting on a long evolutionary branch within the family Chiroteuthidea. The tentacle clubs of its relatives are elongated, but none of them have abandoned suckers. None of them undulate independently. Grimalditeuthis took a family trait and pushed it off a cliff. The evolutionary logic is elegant in a terrible way. In shallow water, tentacles are grabbers. They seize prey and pull it toward the beak. But in the deep midwater, where prey is scarce and energy is everything, a grabbing tentacle only works if something is close enough to grab. Grimalditeuthis turned the tentacle into an advertisement instead. It broadcasts a signal, movement, light, the appearance of something alive and edible, and waits for the response. The predator became a trap. The tentacle became the sign on the door. Only about 20 specimens have ever been examined in detail. Its distribution appears cosmopolitan in deep water, but encounters are so rare that we know almost nothing about its population structure, its mating behavior, or how the detached tentacles eventually reattach. If they do at all. Every answer generates another question, which is itself a signature of an organism in the middle of an evolutionary experiment that hasn't finished running, tentacles that hunt without a body. But the next creatures are losing something more fundamental than appendage function. They are losing their eyes. And you can watch it happening across living species, like watching evolution in time-lapse. Number 3, Bythograeid crabs, the blind vent crabs. Evolution builds. That is the story most people know. Eyes get sharper, brains get bigger, limbs get faster, but evolution also dismantles. When a structure stops providing an advantage, maintaining it becomes a cost. Cells need energy to grow eyes. Genes need to be maintained to code for photoreceptors. If you live in permanent darkness, every calorie spent on an eye you cannot use is a calorie not spent on something that matters, so the eyes go. The family Bythograeid, established by Williams in 1980, contains crabs found exclusively at hydrothermal vents. 6 generi, roughly 14 species, spread across vent systems in the Pacific, the Atlantic, and the Indian Ocean. What makes them extraordinary is not that they are blind, it is that different species are at different stages of going blind. You can line them up and watch the progression. Bythograeid still has compound eyes. They are reduced, fewer ommatidia than a surface crab, but they work. It can detect light. It can sense changes in brightness. It cannot form an image. One step further, Bythograeid has vestigial eye tissue. The structures are physically present, but there are no photoreceptor cells inside them. The eyes exist as architecture with no function. Then, Ostinograeae williamsi, from the western Pacific. Its eye stalks are smooth, blank. There is no eye structure at all, just a rounded nub where an eye used to be. This is not a theoretical reconstruction, these are living animals. You can collect all three from vent fields today. Each one shows you a different frame of the same film. The pattern correlates with depth and darkness. Species at deeper, darker vents show more complete eye loss. And the degeneration is not happening in isolation. As eyes disappear, something else is growing. Chemosensory setae, chemical detecting hairs on the legs and antennae, are increasing in density. Sagonczak and colleagues documented in 1993 that Bythograeid thermodron orients toward sulfide plumes using chemical sensors rather than vision. It navigates by smell, not by sight. The senses are being traded. One system is being shut down and another ramped up, in real time, across a family of living organisms. Molecular phylogenetics by Mateos and colleagues in 2012 confirmed something even more striking. Eye loss evolved independently in at least two separate lineages within the family. This is not a single event radiating outward. It is convergent regression, different branches of the same family arriving at the same solution from different starting points. The family diverged from shallow water ancestors roughly 50 to 65 million years ago, but the eye loss itself may be much more recent. And the evidence for its recency is written into every individual crab's development. The larval stages of these crabs are planktonic, they can see. The eyes develop normally in the larvae, and then degrade after settlement on the vent. Each individual crab recapitulates the evolutionary process during its own lifetime. Born with sight, losing it as it grows, living the rest of its life blind. The developmental program that once built a functional eye is still running, it just stops midway through, a little earlier in each generation, a little more degraded in each species. They swarm vent openings at densities exceeding 500 per square meter. They feed on bacterial mats, vent mussels, and tube worm tissue, mid-level predator scavengers in the vent food web. Their hemocyanin is modified for oxygen transport in hypoxic vent water. They tolerate temperature ranges from 2 to 40 degrees Celsius, and when a vent field dies, they die with it. Genetic isolation between vent populations drives further divergence every time a chimney goes dark. No migration, no gene flow, just drift, divergence, and the slow erasure of structures that no longer earn their keep. The crabs are losing their eyes. But the next creature evolved something in the opposite direction, something unprecedented in the animal kingdom. Armor plating, made of iron. Number 4. Chrysomallon squamiferum, the scaly-foot snail. 2,420 meters down on the central Indian Ridge, at the Kyrai hydrothermal vent field, a team of researchers in 2001 pulled up a snail that should not exist. Its shell was black. Its foot was covered in hundreds of small overlapping scales. And when they analyzed the composition, they found iron sulfide, greygut, and pyrite. The snail was wearing metal armor. Chrysomallon squamiferum is the only animal on Earth known to incorporate iron sulfide into its exoskeleton. The scales on its foot, called sclerites, are arranged in rows like chainmail, each one mineralized with iron compounds precipitated from the vent water. The shell itself has 3 layers. The outer layer is iron sulfide. The middle layer is a thick organic periostracum. The inner layer is aragonite, a form of calcium carbonate. Mariana and colleagues described this architecture in 2003 and noted it was completely unprecedented in the animal kingdom. No other mollusk, no other invertebrate, no other vertebrate, nothing else on the planet does this. But it is not passive. Chen and colleagues demonstrated in 2015 through transcriptomic analysis that the snail actively mediates the mineralization process. It does not just sit in iron-rich water and accumulate metal. The organism controls where and how the iron is deposited. Specific genes are upregulated in the tissues that produce the sclerites. The iron sulfide is laid down in a structured, repeatable pattern. This is biological engineering at the molecular level. 3 populations exist: Chirae, solitaire, and lonqui, each separated by hundreds of kilometers of bare ocean floor with no hydrothermal activity. The Chirae snails have black iron sulfide shells. The solitaire snails have white shells with no iron sulfide at all. Same species, same genome, different armor. The difference is water chemistry. Solitaire's vent fluids do not provide the dissolved iron and sulfide needed for mineralization. What you are looking at is the same genetic blueprint producing radically different phenotypes based on local environment. And the 3 populations are diverging independently. Nakamura and colleagues are tracking genetic drift between them in real time. The divergence is measurable. It is happening now. Yao and colleagues at MIT modeled the 3-layer shell in 2010 and found it outperforms conventional engineered armor at absorbing and dispersing impact energy. The sclerites on the foot may serve dual purposes: armor against predatory crab attacks and structural support for the enlarged foot. The crabs we just discussed, the Bythograeids, are among the predators the scaly-foot snail likely evolved its armor to resist. The arms race is playing out across species, across vent fields, in the same ecosystem right now. The snail hosts endosymbiotic bacteria in an enlarged esophageal gland. Its digestive system is largely vestigial. Like the tube worms we are about to meet, it has outsourced nutrition to chemosynthetic microbes. It grows to approximately 4.5 centimeters in shell diameter, large for a deep-sea gastropod. Population size at each field is estimated at a few hundred to a few thousand individuals. In 2019, the IUCN listed Chrysomallon squamiferum as endangered. It was the first deep-sea invertebrate ever assessed as threatened. The reason, the lonqui vent field, home to the 3rd population, falls within a mining exploration license area granted by the International Seabed Authority. If that license converts to extraction, one of 3 known populations, 1/3 of the species' evolutionary potential, is eliminated, not reduced. Eliminated. 3 populations is already the minimum for maintaining genetic diversity in an isolated species. Remove one and the margin of survival narrows to a razor edge. Iron-plated armor is one way to survive the deep. The next creatures took the opposite approach. They gave up their entire digestive system and thrived. Number 5, Riftia pachyptila. Giant tube worms. In 1977, the submersible Alvin descended to the Galapagos Rift and found something that rewrote the rules of biology. Rising from the volcanic rock around a hydrothermal vent were forests of bone-white tubes, each one capped with a bright, blood-red plume. Some stood over 2 meters tall. They swayed in the shimmering vent current like flowers in a field. They were animals, and they had no mouths. Riftia pachyptila has no mouth, no stomach, no intestine, no anus. The entire digestive tract, the system that every complex animal on the planet uses to process food, is absent, gone, not reduced, not vestigial, structurally and functionally eliminated. An adult tube worm cannot eat. It is not that it chooses not to eat or that its diet is specialized. It physically cannot ingest anything. The structures required for feeding do not exist in its body. What it has instead is a trophosome, a specialized organ that fills most of the worm's body cavity, packed with billions of chemolithium autotrophic bacteria. These bacteria oxidize hydrogen sulfide drawn from the vent water and use the energy to fix carbon dioxide into organic molecules. The worm absorbs those molecules directly. It does not eat, it farms. Felbeck demonstrated the symbiosis in 1981, and the discovery fundamentally changed our understanding of where complex life can exist. Before Riftia, the assumption was absolute. All complex animal ecosystems ultimately depended on photosynthesis. The sun was the base of every food web. Riftia proved that wrong. Life could run on chemistry alone. The blood is remarkable in its own right. The hemoglobin has 3 distinct binding sites. It can simultaneously carry oxygen and hydrogen sulfide without being poisoned by the sulfide, a biochemical feat that no other known hemoglobin achieves. In any other animal, hydrogen sulfide binds to hemoglobin and blocks oxygen transport. That is how sulfide kills. Riftia's hemoglobin carries both molecules on separate sites, delivering the oxygen to the worm's own tissues and the sulfide to the bacterial farm in the tryphosome. The hemoglobin floats freely in the blood, extracellular, not packaged in red blood cells. That bright red plume at the top of the tube is a gill-like organ called the obturacular plume. It is how the worm absorbs dissolved gases from the water. When it senses danger, the approach of a crab, a shift in current, the plume retracts into the tube in a fraction of a second. That retraction reflex is itself an evolutionary response to predation by the Bythograeid crabs from section 3. The arms race between tube worms and vent crabs is ancient, direct, and ongoing. Here is the evolutionary detail that matters most. The larvae can eat. They hatch with a functional digestive system. They swim freely in the water column, planktonic, dispersing on deep-sea currents. When they settle on a vent surface, they acquire their symbiotic bacteria from the environment, not from their parents. Nussbaumer and colleagues showed in 2006 that every generation must be reinfected. The bacteria are not inherited. They are recruited from the surrounding water and sediment. Once the bacteria establish inside the tryphosome, the digestive tract degenerates. The larvae literally lose their guts during development. Each individual worm replays the evolutionary history of its lineage, born with the capacity to eat, then abandoning it in favor of a better system. They grow at up to 85 centimeters per year. The fastest growing marine invertebrate on record, a new vent can go from bare rock to dense tube worm forest in under 3 years. But when the vent dies, so does the colony. They cannot detach, they cannot relocate. Every tube worm forest is a one-generation bet on the longevity of its heat source. Genetic studies by Black and colleagues in 1998 showed high gene flow between populations at different vent sites along the same ridge system, suggesting that larvae disperse over hundreds of kilometers. The symbiosis is estimated to have evolved approximately 100 million years ago, based on molecular divergence from non-symbiotic vestimentiferans. Carbon fixation rates in dense colonies rival surface phytoplankton. Per square meter, a tube worm forest is one of the most productive ecosystems on Earth, and it runs on sulfide and heat instead of sunlight. Related species have since been found at cold seeps, whale falls, and sunken wood across the ocean floor. The chemosynthetic symbiosis evolved once in this lineage and then radiated into every reducing habitat the deep sea offers. It is one of the most successful evolutionary innovations in the history of marine life, and it started with giving up the ability to eat. Tube worms traded their guts for bacteria and thrived. The next creature is trading something else, its skeleton, and scientists believe it is approaching the absolute physical limit of what a fish body can endure. Number 6, Pseudoliparis swirei, the Mariana snailfish. 7,966 meters beneath the western Pacific in the Mariana Trench, Geringer and colleagues collected a small, translucent fish in 2014. They formally described it in 2018. They named it after Herbert Swire, an officer aboard HMS Challenger when the ship first sounded the trench in 1875. Pseudoliparis swirei, the deepest living fish ever recorded. Cameras have observed it at 8,178 meters. At that depth, the pressure exceeds 800 atmospheres. Roughly 1,000 times what you experience standing at sea level. Its skull is not fully formed. The bones are thin, flexible, and unfused. Parts of the cranium are cartilaginous rather than ossified. This is not a developmental defect. A rigid skull would crack under the pressure differential. The flexible cranium bends instead of breaking. It is an adaptation, not a failure. The body is translucent and gelatinous, tadpole-shaped. Minimal musculature. It looks fragile, almost liquid. The skin has no scales. Every structural reduction serves the same purpose: decrease density, decrease rigidity, decrease the number of things that can shatter under compression. The fish is dissolving itself to survive. Inside every cell, the molecule keeping the fish alive is trimethylamine N-oxide, TMAO. It is an osmolyte that prevents proteins from deforming under extreme pressure. Without it, enzymes buckle, membranes collapse, and biological chemistry ceases to function. TMAO props everything open. It stabilizes the molecular machinery against forces that would crush it flat. But T-MAO itself is not free. At higher and higher concentrations, it begins to interfere with cellular processes. Yancey and colleagues predicted in 2014 that at approximately 8,200 meters, the concentration of TMAO required to stabilize proteins would itself become toxic to the cell. That is the wall. The theoretical depth limit for any fish, not a physical barrier, a chemical one. Pseudoliparis swirei lives within 200 meters of it. Evolution has pushed the vertebrate body plan almost as far as biochemistry allows. There may be nowhere deeper for a fish to go. CT scans reveal that the otoliths, the ear stones used for balance and orientation, are proportionally larger than in shallow water relatives. In total darkness, with no visual reference, balance depends entirely on these structures. Evolution enlarged them, compensation again, lose one input, amplify another. It belongs to the family Leporidae, snailfish. And something remarkable has happened in that family. At least 7 species have been documented below 6,000 meters, each in a different trench. The Mariana Trench has its snailfish. The Kermadec Trench has its own. The Japan Trench has its own. Each population is genetically isolated. Each is evolving independently. The trenches are acting as evolutionary islands, driving speciation in real time. The geographic isolation is absolute. A snailfish in the Mariana Trench cannot swim to the Kermadec Trench. The abyssal plain between them is too shallow, ironically, for an organism adapted to hadal pressure. They are prisoners of depth. And in their prisons, they are diverging. Jameson and colleagues observed them in 2019 using baited landers. They were active swimmers, darting across the frame to snatch amphipods. They are not passive bottom dwellers enduring the pressure. They are the apex predator of the hadal food web. Their primary prey is Hirundellea gigas, the dominant amphipod of the deep trenches, a transparent, boneless, gelatinous fish, and it sits at the top of the food chain in the deepest habitat on Earth. The discovery challenged the long-held assumption that the deepest ocean trenches were too extreme for vertebrate life. They are not too extreme. They are just extreme enough to reshape a vertebrate into something almost unrecognizable, a fish whose skeleton is dissolving under pressure. But even at these depths, the snailfish shares its world with something bigger, older, and diversifying so fast that taxonomists cannot keep up with the paperwork. Number 7. Bathynomus. Giant isopods. Milne Edwards described the genus Bathynomus in 1879 from a specimen pulled from the Gulf of Mexico. Bathynomus giganteus. It was half a meter long and weighed over 1.7 kilograms. A deep-sea relative of the woodlouse in your garden. Except this one was the size of a football. And it had been down there, in the dark, for a very long time. There are roughly 20 recognized species in the genus today, and the number keeps climbing. Bathynomus yucatanensis was named by Huang and colleagues in 2022. Bathynomus vaderi was described in 2023 from Vietnamese waters. The taxonomy is struggling to keep pace with the biology. Molecular phylogenetics by Shi and colleagues in 2019 revealed cryptic diversity, species that look nearly identical externally but are genetically distinct. We have been lumping together animals that evolution has been quietly separating for millions of years. The splits are real, the divergence is measurable, and it is ongoing. The gigantism itself is a product of the deep. Cold water slows metabolism. Low predation pressure removes the survival cost of being large. Extended fasting between meals selects for organisms that can store energy efficiently in a bigger body. A specimen in Japan's Toba Aquarium refused food for over 5 years, from 2009 to 2014, before dying. 5 years without eating? The metabolic depression these animals can achieve is almost inconceivable. A large body with a slow metabolism becomes a battery. It stores enough energy to wait for the next whale carcass, the next dead fish, the next meal that might not come for months. Gigantism is not excess. It is insurance. Their compound eyes are the largest of any isopod. Over 4,000 ommatidia per eye, tuned for detecting what little light exists at depth. The exoskeleton is thick calcium carbonate and chitin, forming armored dorsal plates that interlock like medieval plate armor. When threatened, the isopod curls into a ball, conglobation, sealing itself inside its own armor. the same defensive reflex its terrestrial ancestor the woodlouse uses when you poke it in the garden. That behavior has been conserved across hundreds of millions of years of evolution through an ecological transition from land to the deep ocean floor. The body changed. The reflex did not. Reproduction is slow and conservative. Females carry eggs in a marsupium, a brood pouch on the underside of the body. The eggs are few but large. There is no larval stage. Juveniles emerge as miniature adults ready for the seafloor from the moment they leave the pouch. This is direct development, and it is typical of deep-sea crustaceans. Planktonic larvae would drift into the open water column and starve. Direct development keeps the offspring on the bottom, near food, from the start. Geographic isolation drives speciation. Atlantic and Pacific species are more genetically divergent than species within the same ocean basin. Each basin is its own evolutionary arena. The deep water passages between oceans act as barriers, not because the water is impassable, but because the ecological conditions on either side have diverged enough that populations adapted to one basin cannot compete in another. The discovery of new species from Vietnam and the Gulf of Mexico in consecutive years suggests that diversity hotspots exist in regions we have barely surveyed. Southeast Asian waters in particular may be harboring an entire radiation of Bathynomus species that no one has named yet. The genus has also become a pop culture phenomenon: giant isopod plush toys, themed restaurants in Japan and Taiwan, viral social media posts. The public fascination makes sense. It is a creature that looks like an alien, lives like a zombie, and is currently speciating faster than we can catalog. New species appearing in real time, an evolutionary radiation we are documenting as it unfolds. The last creature on our list found a different way to exploit the deep sea entirely. It did not lose a structure or build one. It outsourced the most fundamental act of parenthood to a volcano. Number 8. Bathyraja spinosissima. The volcanic egg skate. Beebe and T. Van described Bathyraja spinosissima in 1941. A large skate, up to 1.5 meters in total length, flat, diamond-shaped, with a long tail studded with rows of thorny spines. It feeds on benthic invertebrates and small fish buried in sediment, detecting them through electroreception, the ampullae of Lorenzini, the same electrical sense that sharks use, inherited from a shared ancestor hundreds of millions of years ago. It lives at depths exceeding 2,900 meters, deep enough that the water temperature hovers just above freezing. And in cold water, developing embryos grow slowly. Very slowly. Deep-sea skate egg cases have been estimated to take anywhere from 5 to over 15 years to hatch. A decade and a half in a leathery case on the ocean floor, developing one cell division at a time. In 2018, Salinas de Leon and colleagues published a discovery that changed how we think about deep-sea reproduction. Using the ROV Hercules at the Galapagos Rift, they documented 157 skate egg cases clustered around hydrothermal vents at 1,660 meters depth. The egg cases were not randomly distributed, they were concentrated near warm vent outflows. The water near the eggs was 2 to 3 degrees Celsius warmer than the surrounding ambient temperature, a small difference by human standards. But for an embryo that might otherwise take a decade to develop, those extra degrees translate to significantly faster cell division, faster protein folding, faster organogenesis, significantly faster hatching. A reproductive advantage that could mean the difference between producing one generation per decade and producing two. The researchers' hypothesis was direct. The skates are deliberately selecting vent-adjacent sites for egg deposition to accelerate embryonic development using geothermal heat. This is not a passive coincidence. The egg cases were found in statistically significant clusters around thermal anomalies. The skates are choosing the hotspots. This was the first documented case of any marine vertebrate using volcanic heat for egg incubation. The parallel that jumped immediately to the surface was the megapode birds of Australia, malleefowl, that bury their eggs in geothermally heated soil near volcanic vents to speed up incubation. Convergent evolution separated by hundreds of millions of years of vertebrate history and an entire medium, air versus water. Different animals, different continents, different ecosystems. Same solution to the same problem. Development takes too long in the cold, so find a heat source and exploit it. Each egg case is dark, leathery, and rectangular. sometimes called mermaid's purses. Each one contains a single embryo. Females produce few eggs per reproductive cycle. The strategy is high investment per offspring, low numbers per generation. Every egg matters. Every embryo is a significant fraction of a female's lifetime reproductive output, and placing those eggs next to a heat source that shaves years off the incubation period is an advantage that natural selection would amplify aggressively across generations. The skates that choose vents produce offspring faster. Those offspring inherit the vent-seeking behavior. The behavior spreads, the population shifts. This is behavioral evolution in action, documented in the present tense. The Galapagos Rift is the same location where Riftia pachyptila was first discovered in 1977. The same vents that feed tube worm forests are serving as incubators for skate embryos. The ecological connections run deeper than anyone initially mapped. Tube worms feed on the chemistry. Crabs feed on the tube worms. Snails armor themselves against the crabs. And skates use the heat to accelerate the next generation. The vent is not just a food source. It is an engine for an entire ecosystem's evolutionary trajectory, but those connections also mean vulnerability. If the vent field is disrupted, by tectonic shift, by mining, by any alteration to the hydrothermal flow, the nursery is destroyed. The eggs cannot be moved. The embryos developing inside those cases are committed to their volcano. There is no plan B. Ongoing research is using temperature loggers placed next to egg cases to measure the exact thermal benefit and estimate how much incubation time is saved. The species is poorly studied outside the Galapagos. Its full depth range and global distribution are not well documented. Other deep-sea skate species may be doing the same thing at vent sites we have not yet surveyed. This may not be one species innovation. It may be a convergent strategy across an entire family of deep-sea rays. We simply do not know yet. The ocean is too large and the cameras are too few. From an octopus that lost its ink to a skate that found a volcano nursery. 8 creatures. 8 different experiments. All of them still running. The deep sea covers more than 60% of Earth's surface. Less than 0.01% of it has been directly observed by human eyes or cameras. Hydrothermal vents are geologically ephemeral. They open, they run for decades or centuries, and they die. When they die, every species that cannot disperse dies with them. The populations that survive at other vent sites are genetically isolated. They drift, they diverge, they become something new. It is the same process that produces unique species on islands, except the islands are chimneys on the ocean floor, separated by kilometers of barren abyssal plain instead of open water. Each vent field is its own evolutionary pressure cooker. Each trench is its own archipelago. The geography of the deep sea is a geography of isolation, and isolation is the engine of evolution. The pattern that connects every creature in this video is not survival. It is modification under pressure, in some cases, literally. Regressive evolution is dismantling structures that cost energy to maintain. Eyes in the Bythograeid crabs, digestive tracts in Riftia, ink sacs in Vulcanoctopus. And compensatory evolution is filling the gaps. Chemoreception replaces vision. Bacterial symbiosis replaces digestion. Touch replaces sight. Enlarged otoliths replace spatial orientation by vision. Every loss is paired with a gain. Every subtraction funds an addition somewhere else in the organism's energy budget. The books balance. They always balance. Evolution is an accountant with no sentiment. At the same time, innovation is producing things no engineer would have predicted. Iron-plated armor in a snail. Autonomous hunting tentacles in a squid. Volcanic egg nurseries. A skeleton that dissolves itself to survive pressure. These are not designs. They are solutions that emerge from billions of random mutations filtered through the most extreme selection pressures on the planet. And the solutions keep coming because the pressures never let up. There is no equilibrium in the deep sea. There is only the next adaptation, the next counter-adaptation, the next arms race. The Bythograeid crabs prey on Riftia tube worms. They snip the plumes when the worms extend from their tubes. The tube worm's rapid retraction response is a direct counter-adaptation. The scaly-foot snail's iron armor may have evolved specifically against crab-crushing attacks. MIT's modeling of the 3-layer shell confirmed it dissipates exactly that kind of compressive force. The vent octopus occupies a niche no other cephalopod has ever claimed, a predator in an ecosystem dominated by arthropods and worms. The phantom tentacle squid's lure strategy is an answer to an energy problem. Active hunting costs too much when prey density is measured in organisms per cubic kilometer. Every adaptation triggers counter-adaptations in competitors, predators, and prey. The arms race never stops. And because the entire contest unfolds in darkness, it is fought through chemistry, touch, pressure waves, and electroreception rather than through vision. The deep-sea coevolutionary landscape is fundamentally different from anything on the surface. It may be more intense because the populations are smaller, the habitats more confined, and the selection pressures more extreme per unit area than anywhere else on the planet. All of which raises a question that is no longer theoretical. What happens when an outside force disrupts these ecosystems before the arms race plays out? The International Seabed Authority has granted exploration licenses covering vent fields and abyssal plains across the Pacific and Indian Oceans. Polymetallic nodule mining targets the abyssal sediments that giant isopods and countless other scavengers depend on. Those nodule fields take millions of years to form. Removal is effectively permanent on any timescale that matters to biology. Vent mining targets the sulfide chimneys themselves, the same chimneys where a Vulcanoctopus broods its eggs, where Bythograeid crabs cluster by the hundreds, where Riftia pachyptila anchors its tubes, and where a Bathyraja spinosissima incubates the next generation. The scaly-foot snail's long key population sits inside a licensed mining exploration area. Environmental impact assessments for these licenses face a fundamental problem. You cannot assess the impact on species you have not discovered. And the rate of new species description in the deep sea, roughly 2,000 per year, means that most of what lives in a mining zone has never been cataloged, never been named, never been studied. Sediment plumes from mining equipment can smother filter-feeding organisms across kilometers. Skate egg nurseries would be directly destroyed by chimney extraction. The eggs are fixed in place. Some nations have called for a moratorium. Others argue that terrestrial mining for the same metals causes deforestation, river pollution, and human rights abuses, and that the deep-sea alternative may be the lesser destruction. The precautionary principle says that when the stakes are irreversible and the knowledge is incomplete, you wait. The creatures in this video are evolving right now. The question is whether they will be allowed to continue. An estimated 2 million deep-sea species remain undescribed. At current rates of discovery, cataloging them all would take 1,000 years. ROV and AUV technology is expanding the frontier. Environmental DNA sampling can detect species from water samples without ever seeing them. Full ocean depth landers now reach the bottom of every trench. Autonomous underwater vehicles running AI-powered species detection can survey for months without human operators. The tools exist, the coverage does not. Every expedition returns with new species, new behaviors, new chemistry. Every survey of an unexplored vent field or trench turns up organisms that have been evolving in isolation for millions of years, unknown to science, following trajectories that no one predicted. The deep sea is not a museum. It is not a repository of ancient relics preserved by cold and pressure. It is an active evolutionary frontier, the largest one on the planet, and it is producing new biological solutions faster than we can document them. Climate change is beginning to alter the baseline. Bottom water temperatures are rising. Oxygen minimum zones are expanding. Deep water is acidifying. These changes may accelerate some evolutionary processes and extinguish others. The interaction between mining, climate disruption, and natural evolutionary divergence in the deep sea is completely unstudied. We are modifying an ecosystem we have not finished discovering. The deep sea is not one environment. It is thousands of microenvironments, each vent field, each trench, each abyssal plain a separate chamber in the largest evolutionary laboratory on Earth. The creatures in this video are not surviving despite the crushing pressure, the total darkness, and the toxic chemistry. They are evolving because of it. Isolation plus extreme selection produces the fastest evolutionary turnover on the planet, and it produces solutions that no one predicted. An octopus that gave up its ink to live in a furnace. A squid that sends its tentacles out to hunt alone. Crabs that are going blind in real time. A snail plated in iron. Worms that surrendered their guts. A fish dissolving its own skeleton. Giant bugs splitting into species faster than we can name them. A skate that incubates its young on a volcano. None of them are finished. Every one of these organisms is mid-process, caught in the middle of an evolutionary trajectory that started millions of years ago and shows no signs of decelerating. What they will become in another million years, another 10 million, is unknowable. Evolution in the deep sea does not follow surface logic. It does not build what we expect. It builds what survives. And what survives down there is consistently stranger, more extreme, and more unsettling than anything the sunlit world has produced. The most terrifying creature currently evolving in the deep sea might be one we have not seen yet. The next expedition will go deeper, stay longer, and look harder. And if the history of deep sea biology is any guide, whatever they find will overturn something we thought we knew. It always does.